Before a simple tube of intimate hyaluronic acid gel is allowed anywhere near a pharmacy shelf, it goes through, well, a gauntlet of mechanical stress tests.
Yeah, genetic mRNA tracking, 3D lab-grown tissue testing. It honestly rivals the protocols for advanced aerospace engineering.
Right. And when you see that little tube sitting there next to the hand lotions, you probably just assume it's a basic comfort product. I mean, you
Yeah.
buy it, you use it, you expect it to be safe, and you really don't think twice about the science behind it.
Which is, entirely by design. A localized comfort product is supposed to feel unremarkable in its safety. We expect it to just work.
Exactly.
But proving that a formulation is, like, biologically invisible to the human body, that requires an incredibly intense, highly rigorous matrix of evaluation.
And that evaluation is exactly our mission today. We are exploring the laboratory testing parameters used to evaluate the biocompatibility and biointegration of intimate hyaluronic acid gel formulations.
It's a huge topic.
It really is. And to do this, we're pulling from a massive stack of sources. We've got FDA 510K pre-market summaries, biomedical patents, advanced rheological studies, and some truly cutting-edge research from MatTech Life Sciences on 3D tissue models.
Oh, that MAPIC research is fascinating.
Mind-blowing stuff. So by the end of this deep dive, you're going to know exactly what it took to engineer that seemingly simple product.
To really understand the modern landscape of how these gels are tested, we have to start with the global regulatory baseline. If you are manufacturing an intimate HA gel, it is classified as a medical device.
Wait, a medical device?
Yeah, exactly. And medical devices must pass the ISO 17993 guidelines. This mandates a standard battery of tests that we can basically think of as the big four of biocompatibility.
Okay, let's unpack this. Because, this baseline testing essentially acts as an extreme background check before a new chemical neighbor is allowed to move into a highly sensitive biological neighborhood.
I love that analogy. So the first hurdle in that background check is cytotoxicity. That evaluates whether the gel directly kills living cells.
Right, and they do that in a petri dish usually.
Usually, yeah. Regulatory bodies typically look for the ISO 10993-5 agar diffusion method. They place the gel on a barrier over a layer of living mammalian cells to see if cellular death occurs.
Makes sense.
Then there is vaginal irritation testing. Historically, under ISO 10993-0 and 23, this involves applying the product in a preclinical setting, usually female New Zealand white rabbits, over a 14-day period.
The 14 days, 14 days of continuous exposure just to monitor for macroscopic signs of tissue damage.
Exactly, like erythema or edema, which is basically looking for visible redness and swelling on the tissue surface.
Okay, so that's two. What's the third?
Following that is sensitization, which looks for delayed allergic reactions. This relies on the guinea pig maximization test or sometimes human repeat insult patch tests.
Basically applying the gel to the skin repeatedly over weeks to see if the immune system slowly builds an allergic response.
Right, you got it. And finally, they require acute systemic toxicity testing. They administer the product via injection in murine models, so mice or rats, to guarantee the gel doesn't trigger a whole-body systemic toxic collapse.
Which, okay, that raises an interesting point based on our sources.
Oh.
Because native hyaluronic acid is a molecule that our own bodies naturally produce.
Right, it's everywhere.
Yeah, it's ubiquitous in our skin, our joints, our connective tissues. It's inherently non-genotoxic. It doesn't cause blood reactivity. So it almost seems like overkill to subject native HA to an acute systemic toxicity injection.
Well, what's fascinating here is that they aren't just testing the raw HA molecule. They're testing the entire chemical matrix.
Because an intimate HA gel is a complex recipe.
Exactly. To make it shelf-stable and effective, manufacturers add preservatives like parabens, viscosity enhancers to change the texture, pH buffers, osmolality regulators.
Any one of those could cause a problem.
Right. The FDA requires this rigorous biological screening because any single one of those secondary ingredients, or even just the way they interact with each other, could be the catalyst that triggers cellular damage.
But relying on this standard Big Four baseline, it actually creates a massive regulatory blind spot.
Yeah.
As we were digging through the FDA 510K summaries, we found a Freedom of Information Act case study about an internal hydrating gel that completely failed the standard cytotoxicity test.
Yeah, it triggered a massive false alarm.
Total false alarm.
This specific FOIA case study perfectly highlights the friction between standardized toxicology and localized human biology.
Yep.
So the manufacturer ran the standard ISO cytotoxicity test on L929 murine fibroblast cells.
And those are just standard mouse cells used in labs everywhere, right?
Yep, super common. The results came back showing moderate reactivity, which is a very clinical way of saying the gel caused significant cell death in the Petri dish.
Wow.
I mean, in a vacuum, those results suggest you have a highly dangerous toxic product.
But the mechanism behind why the cells died is where the standard totally falls apart. Intimate gels are carefully formulated to match the naturally acidic environment of the healthy human vagina, which maintains a pH of around 3.8 to 4.5.
Right, very acidic.
But that standard agar overlays cell culture test with the L929 cells. That test is run at a physiological pH of 7.4.
Yeah, and L929 cells are notoriously sensitive to even minor pH fluctuations.
So wait, if the test is designed for a pH of 7.4 and the healthy human vagina is a 4.0, isn't the test completely flawed? Why would regulatory bodies use a test that effectively penalizes a product for being biologically accurate?
It's a great question. When you take an acidic gel formulated for a pH of 4.5 and drop it onto cells that only survive at a neutral pH of 7.4, the cells experience immediate environmental shock.
They just panic.
Basically, the acidic environment nolazes them. The cells break open and die. But they aren't dying because of chemical toxicity. They're dying because the pH of the environment was violently altered.
Which seems completely contradictory. I mean, if you adjust the product to pass the test at a pH of 7.4, you ruin the product's safety for the actual human environment of 4.0.
Exactly. It forces manufacturers into a grueling regulatory catch-22. The FDA and other bodies rely on L929 fibroblasts specifically because they act as a hypervigilant alarm system.
Better safe than sorry.
Pretty much. The underlying toxicology philosophy is that it is infinitely better for a test to overpredict human response than to underpredict it. But the consequence is that manufacturers have to generate mountains of secondary data to formally justify their failed results.
They have to prove scientifically that the cellular death was just an artifact of a pH mismatch in a plastic petri dish, rather than a genuine toxic threat.
Which is incredibly tedious and expensive.
And proving that a rabbit model or mouse fibroblast isn't a one-to-one match for localized human mucosa is exactly why the industry had to pioneer entirely new methods of testing. The older 2D cell lines and macroscopic animal models were just too biologically blunt.
We needed testing parameters that actually reflected human mucosal architecture. And that brings us to the breakthrough research from MatTech Life Sciences.
Oh, this is the 3D tissue stuff.
Yeah. Bioengineers bypassed the limitations of Petri dishes by developing the epivaginal model, specifically designated as the VLC-100FT.
Rolls right off the tongue.
Doesn't it?
Yeah.
They took normal human-derived epithelial cells, seeded them onto polycarbonate inserts, and cultured them at an air-liquid interface.
Here's where it gets really interesting. By forcing those cells to grow at an air-liquid interface, they trick them into stratifying. The lab is literally growing 3D human tissue that forms the exact multi-layered, fully differentiated mucosal barrier found in the human body.
It's incredible. And because it is anatomically and physiologically identical to human tissue, you can apply the HA gels directly to the surface of the model. You completely eliminate the pH artifact that destroys the 2D cell culture.
That solves the massive blind spot.
It really does. And once the gel is applied, scientists measure the tissue's reaction using two highly precise parameters. The first is the MTT assay.
Right, so the MTT assay measures tissue viability by looking at mitochondrial respiration. It basically tracks whether the cellular engines are still running.
Exactly. Are the cells actually breathing?
If the cells are alive and metabolizing, they convert the MTT chemical into a visible purple color. The deeper the purple, the healthier the tissue.
Yeah. And then the second parameter goes beyond just cell survival to test structural integrity. And that's measured by TEER, transepithelial electrical resistance.
So if I'm picturing this TEER test correctly, it's basically checking the biological mortar between the cellular bricks.
That's a perfect way to look at it.
You pass a small electrical current across the tissue. If the cells are healthy, their tight junctions act as insulators. The electrical resistance is high because the current can't easily pass through.
Right, the wall holds strong.
But if the gel is secretly damaging the tissue, those tight junctions dissolve, the cells pull apart, and the electrical current leaks right through the barrier, causing the TEER score to plummet.
And that structural collapse is exactly what they are looking for. To validate this 3D model, The sources detail a massive double-blind study. Researchers tested 55 different test articles, compounds commonly found in feminine care products, including preservatives, contraceptives, and solvents.
And how did the 3D model hold up?
The epivaginal model flawlessly identified known human irritants. For instance, when benzalkonium chloride or nonoxynol 9, a common spermicide, were applied, the tissue experienced a greater than 50% reduction in both MTT cell viability and tear electrical resistance.
The 3D tissue literally broke down in the presence of harsh chemicals, but it cleared the safe ingredients without issue.
Exactly.
And it did this with vastly higher predictive accuracy for human biology than a 14-day rabbit test ever could.
Yeah, the leap in accuracy is huge, but having intact, surviving human cells is really only the foundation of biointegration.
Oh, right, because of the microbiome.
Exactly. You can have perfect tear scores, but if you introduce that HA gel into the body, it has to coexist with the invisible ecosystem living on top of those cells.
Which is largely governed by lactobacillus species, specifically Lactobacillus crisatus and Lactobacillus inmators. I mean, these are the microscopic good guys.
They are the heavy lifters.
Yeah, they consume glycogen and produce lactic acid, which is what maintains that low 3.8 to 4.5 pH. It's a self-sustaining protective loop.
And that protective loop is the body's primary defense mechanism against infection. If a new HA gel acts as an antimicrobial and kills off the lactobacillus populations, the local pH spikes.
The environment becomes alkaline.
Right, which essentially rolls out the red carpet for opportunistic pathogens.
And so to ensure the gel doesn't disrupt this ecosystem, laboratories conduct zone of inhibition testing. They take agar plates and culture them with the healthy lactobacillus microflora.
And on separate plates, they culture known pathogens like Candida albicans, which is responsible for yeast infections, or Gardnerella vaginalis.
Then they apply the HA gel directly to these plates to monitor bacterial growth. The gel must prove that it neither inhibits the healthy bacteria nor inadvertently acts as a food source that accelerates the growth of the pathogens.
Maintaining that microbial balance is critical, but advanced diagnostics have pushed testing even further.
Because it's not just about the bacteria.
No. Scientists recognize that a gel might not kill the tissue, and it might not kill the bacteria, but it could still be causing microscopic chaos beneath the surface. To detect that, they use a molecular diagnostic tool called QNPA.
Okay, so QNPA stands for Quantitative Multiplex Nucleus Protection Assay. The word multiplex is really the key here, isn't it?
It absolutely is. They aren't just looking for one single reaction. They are simultaneously extracting and measuring the mRNA levels of 25 different genetic pathways at the exact same time.
That's intense.
If we connect this to the bigger picture, they are essentially listening in on the cellular communication network. When tissue is stressed, even before a single cell dies, it starts pumping out specific mRNA markers. The QNPA test specifically isolates markers like TLR4, which is a pattern recognition receptor, alongside IL-1 beta and CXCL8.
And if those specific markers show up, it means the living cells are sending out microscopic SOS signals. I mean, TLR4, IL-1 beta, CXCL8, these are all chemical flags indicating that the immune system is being activated. The tissue is mounting a silent inflammatory response.
And mucosal inflammation is dangerous precisely because it is often completely asymptomatic. A patient won't feel redness or swelling.
But on a molecular level, things are going crazy.
Exactly. The immune system is rushing macrophages and T cells to the mucosal surface to fight off what it perceives as an invader. And in doing so, it inadvertently provides a target-rich environment for sexually transmitted infections, which often hijack those exact inflammatory cells to enter the body.
Wow. So testing the mRNA through QNPA guarantees that the HA gel is truly biologically invisible to the host immune system.
It's the ultimate stealth check.
So we have verified the cellular viability with the MTT assay. We've proven structural integrity with TEER. We've ensured the lactobacillus microbiome thrives. And we've used QMPA multiplexing to confirm there are no silent genetic alarms going off. From a biological standpoint, the gel is safe.
But that still leaves the physical mechanics.
Right, because when a human being actually uses an intimate hyaluronic acid gel, it has to physically perform under friction and pressure.
The transition from biology to biophysics brings us to the science of rheology. Rheology is the study of how complex materials flow, deform, and respond to applied forces.
And hyaluronic acid is a viscoelastic polymer network.
Right. So to quantify how that network behaves, scientists measure two highly specific metrics, the elastic modulus, known as G', and the viscous modulus, known as G'.
Okay, I like to think of this biointegration like the engineering behind a high-end memory foam mattress.
Oh, that's a good way to put it.
Right, because it needs the right elastic modulus, the G prime, to hold its shape, provide structural cushioning, and bounce back when physical stress is applied.
Yes, exactly.
But it also requires the right viscous modulus, the G double prime, so it can flow, adapt to the contours of the body, and actually be extruded smoothly out of a tube. If the G prime is too high, the gel is rigid, like trying to sleep on a rubber tire.
And if the G double prime is too high, it lacks structural integrity, turning into a watery puddle that immediately dissipates.
The balance between G prime and G double prime dictates the shear thinning behavior of the gel.
So laboratories run oscillatory strain sweeps, applying mechanical stress to the gel to ensure the molecular cross-linking adapts dynamically. They rigorously test the extrusion force required to push the gel through an applicator, ensuring it transitions from a stable gel in the tube to a smooth, flowing layer upon application.
But physical integration isn't just about how the gel responds to movement. It's also about how it interacts with cellular hydration. And that brings in the testing parameter of osmolality.
Osmolality measures the concentration of dissolved particles within the gel compared to the natural fluids of the human body. It is arguably one of the most critical, yet frequently overlooked, safety parameters in consumer formulation.
Why is it so often overlooked?
Well, nature always seeks equilibrium across a cellular membrane. If an intimate gel is formulated to be hyperosmolar, meaning it contains a drastically higher concentration of dissolved particles than the surrounding human tissue, osmosis will force water to move across the barrier to dilute the gel.
It's the exact same mechanism as putting a slice of cucumber into a highly concentrated salt brine.
Yes, perfect example.
The brine has a higher particle concentration, so it violently pulls the water out of the cucumber cells, causing the cucumber to shrivel up and lose its structural integrity. If you introduce a hyperosmolar gel into the body, it literally dehydrates the living tissue from the inside out.
Is ripping water out of the epithelial cells and compromising that protective barrier we just tested.
To prevent this osmotic damage, testing parameters strictly evaluate the formulation's concentration.
Right. And while the World Health Organization currently accepts interim osmolality levels up to 1,200 milliosmoles per kilogram for certain lubricants, the ideal, highly rigorous testing standard demands an osmolality below 380 milliosmoles per kilogram.
Which ensures the formulation perfectly matches the isoosmotic balance of human plasma and mucosal secretions.
Exactly. And the mechanical testing goes even further to measure how long the gel actually stays in place. Bioadhesion testing evaluates the physical grip of the hyaluronic acid network.
Right. Our sources describe these ex vivo tests where laboratories take actual vaginal mucosal tissue from pigs or cows, apply the HA gel, and then submerge the entire tissue block in simulated vaginal fluid at exact human body temperature.
Then they mount that tissue in a centrifuge device and spin it at 500 RPMs.
500 RPMs, that's intense.
They are purposefully subjecting the gel to extreme fluid washing and physical shear stress to calculate the exact duration the HA polymer network can stay adhered to the mucosal surface before breaking down.
And alongside that, they run volumetric swelling tests using specialized QIA quick columns to monitor how the cross-linked HA network absorbs biological fluids over time across different pH levels. I mean, the sheer volume of biophysical data required is staggering.
It really is. When you look at the final formulated product, you are looking at an incredibly delicate equilibrium. Formulators are simultaneously balancing the molecular weight of the hyaluronic acid chains, the density of the cross-linking, the rheological G' and G double prime values, the pH buffers, and the osmolality.
And they are doing all of this while guaranteeing that the matrix doesn't trigger a microscopic mRNA immune response or starve a localized bacterial ecosystem.
It's a massive balancing act.
So the next time you find yourself walking down the pharmacy aisle and you spot a simple, unassuming tube of personal hyaluronic acid gel, you'll understand the reality. Getting that product onto that shelf required navigating the flaws of murine fibroblasts, leveraging 3D lab-grown human tissue, decoding multiplex genetic mRNA signals, and calculating precise rheological viscoelasticity.
It is far from just a basic commercial cosmetic. It is an absolute triumph of bioengineering.
It really is.
And honestly, it leaves us with a pretty big thought. If 3D human tissue models and genetic mRNA tracking are proving this accurate for testing topical intimate gels, how long will it be before these localized, lab-grown testing parameters completely replace live animal testing and systemic clinical trials for all pharmaceutical development?
That's a massive shift.
We might literally be looking at the dawn of hyper-personalized intimate medicine.